del día de hoy. Gracias a todos los que están presentes, a los que están presentes en Zoom y en el canal de YouTube. Y realmente es un gran privilegio para mí presentar al doctor Tony Hunter el día de hoy. Aquellos que iniciamos nuestra carrera en señalización hace algún tiempo, nos tocó seguir de cerca los descubrimientos que siguieron al descubrimiento principal, que fue la fosforilación en tirosinas, y cómo eso cambió nuestra manera de ver cómo una célula responde a las señales metagénicas y cómo favorece eso la transformación celular. Y ayer Tony les dio una historia muy bonita a los chicos en la licenciatura, y bueno, va a hablar desde la fosforilación en tirosinas hasta la histidina, que es lo que está estudiando el día de hoy, y realmente es un placer para mí presentarlo. Tony obtuvo un grado de licenciatura en bioquímica por la Universidad de Cambridge. Después obtuvo una maestría también en Cambridge, y su doctorado también en Cambridge, siempre trabajando en bioquímica de proteínas. Después, una vez que terminó su doctorado, se quedó también en la Universidad de Cambridge, en el Christ's College, como investigador independiente junior por 3 años, y después se cambió al Salk Institute a hacer una estancia postdoctoral, donde estuvo también 3 años, en el grupo del doctor Walter Eckhart, donde estaban precisamente estudiando cómo los virus podían inducir transformación celular. Regresó a la Universidad de Cambridge, al Christ's College, otra vez como investigador independiente, pero en el 75 decidió regresar al Salk Institute, y desde ese entonces hasta la fecha es investigador en el Salk Institute. Empezó evidentemente como profesor asistente, después con el tiempo se hizo profesor adjunto en la División de Biología de la Universidad de California en San Diego, más tarde también profesor adjunto del Departamento de Farmacología en la Universidad de California en San Diego. Se promovió a profesor en 1982. Después fungió como director del Cancer Center del Salk Institute de 2016 al 2017, y desde 2011 fue reconocido como investigador en nombre del doctor Renato Dulbeco hasta el presente. Y bueno, Tony tiene muchísimos premios y reconocimientos. Me llevaría muchísimo tiempo leerles todos, pero si les menciono los más recientes, ganó en el 2018 el Tag Prize for Biopharmaceutical Sciences. Después recibió el reconocimiento a una vida de investigación por la organización ACCR en investigación en cáncer en el 2022. Recibió también el premio de la Fundación Gelis por su investigación en cáncer también en el 2022, y es un investigador honorario en el College Gonville and Cassius en Cambridge desde el 2022. Sobra decir que ha participado en múltiples comités editoriales de las mejores revistas científicas en el área, en múltiples comités de premiación también, y en comités de supervisión a compañías farmacéuticas a lo largo de su carrera. Tony al día de hoy en el CV que me envió tiene 600 publicaciones aceptadas y otras 10 que están en camino. Todas ellas, otra vez sobra decirlo, en de las mejores revistas en el área. Aunque Tony no manda sus citas, me imagino que son muchísimas. Y realmente otra vez es un privilegio para mí compartir esta tarde con Tony, y el tiempo que ha venido en estos 2 días a compartir su experiencia con los chicos del ESG, porque realmente representa un parteaguas en la ciencia de lo que él descubrió. Entonces, Tony, thank you very much for being here, and it's all yours.
Thank you, Gustavo. That was very kind, although I didn't understand all of it. I sort of got the gist. It's a pleasure to be back here in Cuernavaca and give another lecture in the Frontiers in Genomics series. I last talked here 15 years ago in 2008. So I thought I'd start with a little history. I went through this with the students yesterday, so I'll go through it very briefly now just to bring everyone up to speed about the phosphorylation field. So protein phosphorylation was identified well over 100 years ago now, particularly with the work of Levine and Olsberg, who showed that vitelline, an egg white protein, contains covalently attached phosphate. And they reported this in the second issue of the Journal of Biological Chemistry in 1906. It took another 25 years before they discovered that the phosphate was covalently linked to serine, Lippmann and Levine in 1932. The next phosphoamino acid to be identified in proteins was phosphohistidine, the work of Paul Boyer's group, who showed that a mitochondrial enzyme contains phosphohistidine. And this was the key paper, again in JBC in 1962, where they identified this protein that was labeled in a mitochondrial preparation to which inorganic p32 phosphate was added, which then the mitochondria generated, labeled ATP, which phosphorylated a protein that they showed was the phosphate linkage was very labile in acid and fell off. And it turned out they later showed that this phosphohistidine was the enzyme intermediate in this, it was E. coli succinyl-CoA synthase, here we go, which is an enzyme in the TCA cycle. So this was the first enzyme known to use a phosphohistidine intermediate, but there are several others, as you will see. And then tyrosine phosphorylation was identified in 1979, 1980, with particularly our demonstration that the Ras sarcomavirus transforming protein, VSARC, contains phosphotyrosine when isolated from cells. And I went through the history of the discovery yesterday. I'll just tell you what we were doing. We were trying to identify the amino acid phosphorylated in vitro by the polyomavirus, another transforming virus, middle T antigen, that had been labeled in the presence of gamma p32 ATP, and then hydrolyzed to release phosphoamino acids in 6-normal HCl. And the hydrolysate was then run out by electrophoresis at pH 1.9 on a cellulose thin layer plate with added phosphoserine and phosphothreonine markers. And then the plate was dried and exposed to an X-ray film to detect the P32. And when I put the X-ray film down on the plate overnight and then developed it the next morning, this is what I saw, which you probably can't see anything obvious. There is a faint smudge here, which with a longer exposure turned into this quite dark spot, which didn't migrate with either phosphoserine or phosphothreonine. And I proposed that this might be phosphotyrosine, and subsequently we showed that that was indeed the case. So that was a very brief history of tyrosine phosphorylation. Now, phosphorylation is a widely used post-translational modification for regulating protein activity. It's a reversible modification with a writer enzyme, a protein kinase, that adds the phosphate to the protein using ATP as a donor, and an eraser enzyme, a protein phosphatase, that removes and hydrolyzes the phosphate ester linkage that's created by phosphorylation, releasing free phosphate. Often, so this changes the protein state in several different ways, but one common way that phosphorylation is used is for recognition by a phosphobinding protein, which contains a phosphobinding domain like the Salk homology 2 domain that recognizes phosphotyrosine. This is called a reader. Now, there are over 400 different post-translational modifications, the new ones being identified all the time, largely through the use of mass spectrometry. But protein phosphorylation is the most prevalent PTM in eukaryotes, with as many as 50,000 sites being occupied on proteins in a single cell type. As I'll show in a second, this is a major mechanism for increasing proteome complexity. So a mammalian genome starts with around 20,000 protein coding genes. This can potentially encode about 100 proteoforms by alternative splicing and alternative starts, which is a large number, but perhaps not a large enough number to drive a very complex organism. And it's through the use of combinatorial post-translational modifications that one can generate potentially millions of distinct protein states, each of which will have a subtly different function. And so we think this is a major function of post-translational modifications. And just some numbers here, the cell signaling technology phosphocyte plus database lists nearly 300,000 distinct serine, threonine, and tyrosine phosphocytes. Not many of, very few of these have actually been validated, and so it's not clear whether this is a real number or not. But compare that to the number of ubiquitination sites, simulation sites, acetylation sites that have been identified, or monomethylation sites. And it's clear that post-translational modification is a very important process. And used combinatorially, it clearly has a very complex outcome. Just as an example of the sort of proteins that can recognize these post-translational modifications, here are a few examples of domains, modular domains that are known to recognize phosphotyrosine, the SH2 domain, phosphothreonine, the FHA domain, phosphoserine, the BRCT domain, and then ubiquitin and sumoylation sites can be recognized by modular protein binders, as well as particularly in the nucleus for chromatin regulation, methyl lysine and acetyl lysine can be recognized. So the very large number of phosphocytes in a mammalian cell indicates that probably a large number of writer enzymes, in other words, protein kinases, maybe an equally large number of protein phosphatases to reverse their activity. So I became interested in how many protein kinases there were nearly 40 years ago now, when the first sequences of some of the virally encoded protein kinases like VSark became available through cDNA cloning of the viral genomes. And in the case of these three serine threonine kinases, the cyclic AMP-dependent protein kinase, the cyclic G-dependent kinase, and phosphorylase kinase gamma, the protein, these were sequenced the old-fashioned way at the protein level. So I began to align these sequences by eye. So BB here stands for before blast. So this was done by eye and not by a computer, or rather it was done by my brain, I guess. And it was clear that there were conserved motifs, both in the tyrosine kinases on the top and the serine threonine kinases on the bottom, that were arrayed in a particular order along the length of the catalytic domain, which is around 300 amino acids. And in particular, this domain, the HRDL domain, which contains the catalytic aspartate that promotes phosphate transfer, was different between the tyrosine kinases. It was HRDL, AARN or RAAN, and the serine threonine kinases, where it was RDLKPEN. And so this motif has actually been used subsequently for predicting whether a new kinase-like sequence might be a tyrosine kinase or a serine threonine kinase. So the first sequence alignment came out in a rather unusual place, namely in the Scientific American, in an article I wrote on the proteins of oncogenes in 1984, and then a somewhat more official alignment came out in the Annual Review of Biochemistry. And together with Steve Hanks, who was at the Salk Institute, we began to align all the new catalytic domain sequences, and were able to deduce that there were 12 different subdomain motifs of the sort that I showed you in the last slide. And the catalytic domains all contain these motifs and can either be an entire protein, as is the case for the pKa catalytic domain, or part of a much larger protein, as is the case in the EGF receptor tyrosine kinase. Now, it turns out that these motifs are located in key positions around the catalytic cleft of the protein kinase catalytic domain. This is the first catalytic domain structure from Susan Taylor's group at UCSD. And so you can see the DFG motif is in here, the RDL catalytic aspartate is in here. And then the domain is divided into two parts, if you like, the C-terminal lobe where the substrate binds, which is mostly helical, and the N-terminal lobe, which is mostly beta strand with this C helix here. And you can see there's a bound ATP molecule in this structure. Here's the adenine ring. And here is the gamma phosphate waiting to be transferred onto a substrate, although the substrate is not in this structure. So mutation of these motifs, particularly this lysine here, for instance, or the catalytic aspartate, almost totally abolishes kinase activity. So they're clearly functionally very important. So more and more sequences began to appear largely through PCR-based cloning. And the number of protein kinase-like sequences began to rise almost exponentially in the 1980s, as did the number of tyrosine kinases. And so in this little thought piece that I wrote in 1987, 1001 protein kinases, it looked like there might be as many as 1,000 protein kinases in the kinome, the complete collection of kinase genes in a single organism. So fast forward many years now when complete genome sequences became available, the human kinome turns out only to have around 535 kinase genes, which is quite a large number. But there are organisms like the eucalyptus tree and the rice plant, which have very large numbers of protein kinases. Plants have had several whole genome duplications, and so this is probably the cause of this very large increase. So there are many copies of each kinase. So when the complete draft human genome became available in 2001, we were able to scan the sequence using hidden Markov models, looking for these 12 motifs, and came up with this tree of related eukaryotic protein kinases in 7 major branches. Tyrosine kinases on top, the basophilic kinases down here, including PKA, calcium calmodulin regulated kinase and the CMGC family, which contains the proline-directed kinases like the CDKs and the MAP kinases, and GSK3. So there were 518 in that tree, but in addition, there were a number of atypical protein kinases which have been reported. Not all of these have stood the test of time. The ABC kinases are real. The alpha kinases are real. These are probably not real. And particularly important are the PI kinase-related kinases, which includes ATM, ATR, and DNA PK, ancient, very large kinases that recognize DNA and RNA damage, and mTOR, which is the energy sensor kinase of all eukaryotes. So of the 518 protein kinases, interestingly, about 10% are missing key catalytic residues and are probably pseudokinases in most cases. And where we've looked at them, they appear to have scaffolding functions. They're folded like a real protein kinase, but they lack activity. That was 2002. Now in 2018, Kinome 2.0, about 20 new kinases have been added, but they're all remote or atypical. These include the real casein kinase, FAM20C, a secreted protein kinase in the lumen of the Golgi that phosphorylates proteins that are being secreted on their way out of the cell, and the NME family of histidine kinases that I'll talk about. To oppose the kinases, there are indeed a large number of protein phosphatases, in this case in many different structural families, 10 structural families, in this review by Chen and Manning. The largest family, 107 members, are the protein tyrosine phosphatases and dual specificity phosphatases. And this family here of 1 is one of the phosphohistidine phosphatases that I'll talk about. So the total number of catalytic domains is around 190, about a third of the number of protein kinase catalytic domains, but many of the catalytic domains have regulatory subunits and increasing the total number of distinct protein phosphatase entities. So about 4% of all human genes are protein kinases and phosphatases, so a major functional activity in the cell. Now, my initial interest was how many tyrosine kinases there are. It turns out the human kinome has 90 tyrosine kinases. This was the founding tyrosine kinase Salk. The largest receptor tyrosine kinase family is the EPH family with 14 members. About 58 out of the 90 tyrosine kinases are receptor-like kinases that span the membrane and act as receptors for various ligands. But there are another 32 non-receptor tyrosine kinases, such as the TEC family here, the RJ-able family here, and the JAK family over here. Over half of these are implicated in cancer and other diseases through mutational evidence or overexpression. And this number's a little soft, but probably over a third of human protein kinases are involved in disease. And the majority are involved in cancer, but there are a number of important kinase mutants that cause immunodeficiency, cardiovascular disease, or developmental malformations. Most of these are loss of function mutations. So because of the involvement of tyrosine phosphorylation in cancer, there was a lot of interest in developing small molecule inhibitors in potentially as new types of drug to target different cancers with mutant gain-of-function tyrosine kinases. Over the last 22 years, since Gleevec was approved in 2001, 88 different protein kinase inhibitors or protein and PI3 kinase inhibitors have been approved. 63 of these are directed against activated tyrosine kinases. And here's a timeline that came out in a review by Atwood et al. In 2021, showing the sort of explosion of approval of different tyrosine kinase inhibitors. Some of these are protein drugs, actually. They're monoclonal antibodies that target receptor tyrosine kinases. But there are 88 small molecule kinase inhibitors, which 63 are tyrosine kinase inhibitors. This all started with Gleevec, or generically known as imatinib, the inhibitor of the BCR-ABL oncogene that is the driver of chronic myelogenous leukemia. There are several hundred more in clinical trials, and these two have been approved in the last year or so. Asciminib here is an allosteric inhibitor. Most of these small molecule inhibitors target the ATP binding cleft in one way or another. This is an allosteric inhibitor and can be used in combination with imatinib. And here's a very new one that targets the TYK2 tyrosine kinase involved in inflammatory responses as a member of the JAK family. Okay, so that's the background. I'll start talking about a project that sort of indirectly involves tyrosine phosphorylation, and then move on to talk about histidine phosphorylation in the second half. So we became interested in, I've studied cancer biology my whole career, or nearly my whole career. And I never really focused on a single cancer type. But about almost 15 years ago now, I was persuaded to join a team at Salk that was going to study the role of the stromal cells, the normal cells in pancreatic cancer. See whether we could target those cells which act as aids to the tumor cell. And just to sort of underscore how important it is to study pancreatic cancer, the 1-year survival is less than 20%, and the 5-year survival is 9%. And it's estimated that pancreatic cancer will become the second leading cause of death. I think it's probably not quite there yet. And the main reason is that it's a cancer that's diagnosed too late. It's asymptomatic until it's generally metastasized, which is basically untreatable. So, and even when it's diagnosed, there's no really good targeted therapy. And the reason it's hard to treat is that it's a highly stromal tumor in which nests of tumor cells with invasive, largely myeloid cells like tumor-associated macrophages is surrounded by these stromal cells, largely cancer-associated fibroblasts, which are proliferating and generate a very dense collagen extracellular matrix, which it's really a wound response. The tumor has induced a normal tissue wound response, so that the stromal cells are trying to wrap up this cancer, but cancer is often being called the wound that never heals, because the cancer cells can ultimately win and escape. The other important thing about pancreatic cancer is that it's highly immunosuppressive. And so it's a cold tumor, and cytotoxic T cells are excluded around the periphery of the tumor and do not infiltrate. So as I said, we were interested in the stromal cells, so that's largely the cancer-associated fibroblasts, sometimes called stellate cells, which are sort of tissue-resident cells that monitor the tissue status. And we were interested in what factors these stromal cells make, soluble secreted factors that might act on the tumor cells, and potentially what proteins the tumor cells make that act in trans on the stellate cells. And some of these are already known. So what we identified was a cytokine called LIF, leukemia inhibitory factor, which is a member of the IL-6 family. And I'll just take you through that story, which was published, and talk a little bit about some more recent data. So the way we did this was the work of Yu Shi, a postdoc in my lab, and Ruijun Tian, a postdoc in Tony Paulson's lab, was to culture human stellate cells or cancer-associated fibroblasts in serum-free medium, isolate the medium, conditioned medium, and do proteomic analysis. And we took 2 different human pancreatic tumor cell lines, both driven by mutant KRAS, did the same thing. And we found a large number of secreted proteins, as you can see, in each case. And we focused on the factors that were unique to each cell type. And in particular, as we were studying the stromal cells, we focused on growth factors, cytokines, and chemokines that might act on the tumor cells. And we decided to focus on LIF, member of the IL-6 family, which is also made by these cells, because very little was known about its role in cancer. So we chose LIF for the following reasons. The conditioned medium from CAFs stimulated phosphorylation of STAT3 on tyrosine 705. This is a known cytokine response mediated by the JAK tyrosine kinases. So this suggested there was an active cytokine. We tested LIF directly using antibodies and could see that conditioned medium elicited binding of STAT3 to the LIF receptor and its co-receptor in the tumor cells, and showed that a neutralizing LIF monoclonal antibody blocks STAT3 tyrosine phosphorylation by the conditioned medium. So LIF was of interest for other reasons. It's a stem cell factor. It's used to maintain the pluripotency of ES cells in culture, and its normal function is to promote the implantation of the blastocyst into the placental wall, where it creates an immunosuppressive microenvironment to avoid rejection of the embryo. So these were properties that seemed would be useful to a tumor cell. And then we showed subsequently that the human pancreatic tissue and their fibroblasts contain high LIF levels. So we looked to see, using in situ hybridization with RNA-scope, two-color RNA-scope, where both LIF and the LIF receptor were expressed in a mouse model of pancreatic cancer called the KPC model. It has an activated KRAS and loss of function P53. So if you look at the red signal here of LIF, it's mostly in cells surrounding the blue tumor cells, which express keratin 19. And LIF receptor is largely in the tumor cells, not in the periostin positive stromal cells. So it looked like the activated CAFs around the tumor were making LIF that was acting on the tumor cells to drive tumor progression. So we looked at this more directly in the KPC model by administering either a normal control IgG or the anti-LIF antibody, D25, at 25 mg per kg in the KPC mouse model, and showed that the antibody largely suppressed phosphostat-3 in the nuclei of the tumor cells. So then we carried out preclinical studies in the KPC model to test its potential therapeutic efficacy. In this case, we carried out a pre-treatment with nab-paclitaxel, a microtubule disruptor, gemcitabine, nucleotide metabolism disruptor, and cisplatin, a DNA damaging agent, for 1 week, followed by these are conventional therapies for pancreatic cancer, which really don't work, but that's what's used. And gemcitabine coupled with anti-LIF antibody. And particularly the combination of gemcitabine with the anti-LIF antibody gave a significant increase in survival, about a 2-fold increase in survival of these mice. Not a cure by any means. It's a very hard tumor to treat, but this looked encouraging. What we showed was that LIF blockade in the mouse enhances the response to chemotherapy by upregulating the gemcitabine transporter into the tumor cells, and also by decreasing the number of stem-like tumor cells with stem-like properties. Obviously, that was the mouse. What about in human pancreatic cancer? Through collaborators, we obtained a number of pancreatic cancer tissue samples, different stages from stage 2 to 4. And many of them contained very high levels of LIF as determined by an ELISA assay compared to chronic pancreatitis or normal pancreas. And using Quanterix digital ELISA, we could show that many patients also had high levels of LIF circulating in the serum. Note, this is a log scale here, but not all. Unfortunately, we don't have serum and tumor samples from the same patients. So this was published some time ago now. So the conclusions were that LIF plays a driver role in pancreatic cancer in a mouse model, that LIF levels are elevated in serum and tumor samples in the human disease. And it can also not only serve as a therapeutic target, potential as a biomarker because of elevated levels in serum. So the big question is then, can LIF be targeted in the human disease? And a company called Northern Biologics, which was not associated with, developed another neutralizing LIF antibody and ran it through a phase 1 clinical trial, an all-comers cancer trial, where there were no adverse responses at the highest dose, and one pancreatic cancer patient showed slightly prolonged disease. That drug or antibody has now been taken into a phase 2 combination trial by AstraZeneca. So if LIF receptor interaction and signaling is indeed a potential target, how can one antagonize its function? I've already told you can use an anti-LIF antibody, but in principle, you could use a LIF receptor neutralizing antibody, and there are companies developing these. One could engineer a LIF receptor trap, and one could potentially develop small molecule blockers of LIF-LIFR interaction. And one such molecule has been reported. Now, this was an interesting possibility that my son, Sean, decided to, in his graduate student lab at Stanford, decided to explore. And what he did was, in Jennifer Cochran's lab, was to engineer these 3 domains of the LIF receptor ectodomain by yeast surface display using iterative rounds of mutagenesis. And he identified a molecule that he's called ELIFR-FC with 8 point mutations fused to the human FC that has about a 30-fold increase affinity for LIF. And this was used in a mouse model, which we helped with, to show that it slowed administration of ELIFR-FC, slowed the growth of these human pancreatic cell xenografts in nude mice. So this in principle then is another approach to antagonizing LIF signaling that could be taken forward into a company. So what happens next then? Well, I told you, an immunologically cold tumor. So what AstraZeneca is doing is to combine MSC1 with their anti-PD-1 antibody durvalumab together with gemcitabine and nab-paclitaxel and cisplatin to see if they can improve an immune response. I would say that work in Ron Evans' lab in collaboration has shown that treatment with anti-LIF antibody promotes T-cell killing of KPC cell orthotopic tumors when they're engineered to express an antigen recognized by the T cells that are introduced adoptively. So I think there is potential that antagonizing the immunosuppressive activity of LIF may be useful in pancreatic cancer therapy, and we'll see what happens in this trial. So I showed a picture of my beard dribbling kinases yesterday. Well, this is a picture of my beard dyed purple to raise money for pancreatic cancer. So let's move on to histidine phosphorylation, identified 60 years ago now. It's one of 6 amino acids, in addition to serine, threonine, and tyrosine, that can be phosphorylated. These are the classical phosphorylations. They're very heat stable and implicated in cancer when phosphorylated. These 6 amino acids are all what we call non-canonical phosphorylations. And the phosphate linkages in each case, particularly in arginine, lysine, and histidine, which are phosphorus-nitrogen bonds or phosphoramidate linkages, are heat and acid labile, making them very difficult to study. And we wanted to ask the question whether histidine phosphorylation in particular is involved in cancer, and if so, could it be targeted? So just as a reminder then, this is tyrosine. It's phosphorylated on the phenolic hydroxyl group, forming a phosphate ester that's acid and heat stable. This histidine with its imidazole ring can be phosphorylated at either one position or the 3 position to form a phosphoramidate linkage. Phosphoramidate linkages fall apart in acid, any pH below around 6, and fall apart when you heat the sample. So what do we know about histidine phosphorylation of proteins? Well, many of you will know about the bacterial 2-component systems where a receptor-like protein has histidine kinase activity that phosphorylates a histidine in a receiver domain. That phosphate is transferred onto a aspartate residue in a second protein, usually a transcription factor, switches on transcription. So this is an unusual phosphohistidine phosphoaspartate relay. These 2-component signaling systems have been lost in multicellular eukaryotes. Some of the single-cell eukaryotes have 1 or 2 of these systems. In mammals, we don't have these. We have potentially, though, histidine kinases, NME1 and NME2, which work through a phosphohistidine intermediate, and that phosphate can be transferred onto a histidine in a protein, and we have at least 3 potential phosphohistidine phosphatases. So this could be a reversible post-translational modification system. And I'll tell you about at least one example where there's some evidence that histidine phosphorylation of a site in a potassium channel increases its channel opening. So this is what we were trying to understand. Is it possible for a histidine in a protein to be phosphorylated by histidine kinases using ATP at the 1 or 3 position, which can then be reversed, releasing free phosphate by phosphohistidine phosphatases? Now, the NME1 and 2 enzymes are probably better known as nucleoside diphosphate kinases. They have a housekeeping function of rephosphorylating nucleoside diphosphates using this active site phosphohistidine, which is a 1-phosphohistidine, to regenerate the triphosphate. But there's experimental evidence that the same enzyme intermediate can phosphorylate proteins. So this family of 10 enzymes may actually be dual function. So in addition to NME1 and 2, which have a phosphoenzyme intermediate, and succinyl-CoA synthase that I already told you about, several other enzymes have phosphoenzyme intermediates. And phosphoglycerate mutase, an enzyme in the glycolytic pathway, uses this intermediate that I'll mention. In case you don't believe there's phosphohistidine in proteins, there are actually structures. It's stable enough to do crystallography. So here's the 1-phosphohistidine in NME1. Here's the N1 on the phosphate. And here's the phosphoglycerate mutase intermediate. Here's phosphate on the N3 position of histidine 11. So this is a sort of summary of what we know about histidine kinases, which includes the bacterial 2-component system kinases. Here are some targets that have been identified, including ACLY, the beta subunit of G proteins, and this potassium channel for NME1 and NME2. And these are the potential phosphatases, particularly PHPT1, LHPP that I'll mention, and PGAM5, a distant relative of PGAM. And here are some of PHPT1 substrates, and we've just found that SCS, succinate thiokinase alpha phosphohistidine, is also a substrate for PHPT1. So to come back then to our schema here, I'll just very briefly summarize this system. Mostly the work of Ed Skolnick at NYU, who has studied this channel. It's a calcium-activated potassium channel, especially important in T cells, which is activated upon T cell receptor engagement. And what he showed was that this involves calcium binding to calmodulin, which is resident on the tails, the 4 tails of this tetramer receptor, that leads to phosphorylation on the 3 position of the histidine 358. NME2. This phosphate is removed by PHPT1. And NME2 can be dephosphorylated by PGAM5. And their model is, and there's some evidence, that this histidine is a copper ion coordinates the 4 histidines, closing the channel. And when the histidine is phosphorylated, the copper is dismissed, and now potassium can be released, which allows additional calcium to enter the cell and drive T cell activation. So to summarize all that, we put together this cartoon here. There is histidine phosphorylation in the nucleus of histone H4. There's phosphorylation of channels of the G beta subunit. And as I'll show you, there's phosphorylation of proteins at the centrosome and the spindle poles. The mitochondria has 2 different NMEs associated with it. And it appears that histidine phosphorylation may be important in mitochondria as well. So how can we study this unstable modification? Well, we thought one way would be to generate phosphohistidine-specific antibodies by analogy with phosphotyrosine antibodies that have been very helpful in studying tyrosine phosphorylation. We tried to do this 30 years ago, but failed because our phosphohistidine-containing protein didn't seem to present phosphohistidine. So we waited until 2 groups generated these stable analogs, phosphorylazoalanine analogs of phosphohistidine, where the phosphate is linked to a carbon, a phosphonate linkage in a 5-membered ring with nitrogens in the 1 position or the 3 position. And we immunize rabbits with peptides containing either 1 or 3 PTZA with random adenine and glycine for 5 residues on either side, hoping to get sequence-independent antibodies. And we did get good antibodies that recognized either 1 or 3 phosphohistidine proteins and made monoclonal antibodies. And this was published a few years ago now. Here I'm showing you some immunoblots of pancreatic cancer cell lines coming over from the Pancreatic Cancer Project, where the main proteins we see are phospho-NME1 and 2. And in each case, we have a control lane where this same sample was boiled at 95 degrees, and the histidine is lost, as I told you it would be. So this is always our control. Here we have, and that was with one of our 1P His monoclonals. Here we blotted an E. coli lysate and a 293 cell lysate with 2 of our 3P His antibodies. And you can see that there are many bands, some of which we know, mostly lost upon boiling. So we have 3 unique 1P His and 4 unique 3P His antibodies. We've used them for immunoblotting, immunofluorescence staining, immunohistochemistry, and for affinity enrichment of P-His proteins and peptides for mass spec analysis to identify sites of histidine phosphorylation. And we've done the structures of 5 of these monoclonal antibodies bound to PTZA peptides. I think given the time, I'm going to skip through the structures which are published anyway. I'll just say that for the 1P His antibodies, we know that phosphate binding involves conserved arginine that is similar to the phosphobinding motifs in phosphotyrosine binding domains like the SH2 domain. And for the 3P His antibodies, you can see they all have a positively charged cleft into which the phosphate is drawn. There is a XX-gly motif similar to the P-loop or Walker A motifs in proteins that bind nucleotide triphosphates. That's just a blow-up of what this looks like for this SC44 protein antibody that I'll be using in some of the experiments. So we've used these antibodies to stain cells. Here is one of the 3P His antibodies staining proliferating HeLa cells. And you see some nuclear dots and some cytoplasmic dots. But strikingly, we see in early prophase these two dots, which are the centrosomes that then split to form the spindle poles, suggesting a role in cell cycle progression. And we used immobilized, a mixture of 6 of these antibodies immobilized on beads to pull out phosphohistidine proteins from lysates of 293 T cells that were denatured in 6 molar urea at pH 10, and then diluted and neutralized for pouring over the column. And a control was half the lysate was treated at pH 6 and boiled to discharge phosphate off histidine. And we found around 800 proteins that were enriched twofold. Many of these proteins, this set on top here, were involved in RNA biology, splicing, transcription, RNA nucleoprotein complexes, and about 100 were predicted to be involved in cell cycle progression. So these could be important histidine phosphorylation targets. And we found as sort of controls, if you like, histone H4, NME12, PGAM1, and ACLY, the phosphoenzyme intermediate. Now, this doesn't prove they contain phosphohistidine. We actually need to do site identification. And that's challenging because conventional phosphoproteomics uses strongly acid conditions, and so you can't readily use that. So we developed a protocol in which we use bead immobilized anti-PHIS monoclonals to enrich phosphohistidine peptides from tryptic digest at pH 8, and then subjected them to positive ion mode mass spec analysis. We found a number of sites that were predicted to be above the 95% statistical probability, but I should say that identifying phosphohistidine sites from mass spec is not straightforward, and that it is somewhat controversial. But we did find phosphoglycerate mutase, phospho-H11, so that was gratifying. And some other interesting proteins. And more recently, we've really, I think, pretty much nailed that this splicing factor is phosphorylated on histidine. So this is something we're going to work on. There's no strict motif for histidine phosphorylation, unlike other phosphokinase sites. Lots of leucines, arginines, and lysines. And we think these actually probably are involved in peptide, phosphohistidine peptide stability rather than because they are selected by the kinase, but that waits determination. So how abundant is phosphohistidine in the mammalian cells? Claire Ayers has developed a different method for enriching phosphohistidine peptides and has identified, I think, around 70 phosphohistidine sites. Other mass spec studies using different enrichment methods have also identified many PHIS sites. And using P31 NMR analysis, Muimo's group has been able to detect the 3 PHIS peak here, which is pretty minimal, as you can see, and compared that to the peak you see for the phosphate esters of phosphoserine and threonine on the beta hydroxyl group, and has argued that this means 3 PHIS might be 30% as abundant as phosphoserine and threonine. I suspect that's an overestimate. So the hidden phosphoproteome, as we've called it, could be quite large, but a recent paper has suggested that maybe these were all overestimates. And so I think the jury is probably still out. If you're interested, you should read this paper and see why they criticized all of the previous studies. I mean, our immunoblotting results combined with the fact that we know how specific the antibodies are suggests that really there are a significant number of PHIS sites in cells. So what are the open questions? What cellular functions might be regulated by PHIS phosphorylation, cell cycle, or RNA processing and transcription? How big is the phosphohistidine proteome? I think we have to wait Are there other histidine kinases and phosphatases? Maybe. Is there selective phosphorylation at the 1 or 3 position? We don't know. We suspect 3 is the most likely target because it's more stable. Are all of the sites that are identified functional? Probably not. And how are they decoded? Is phosphohistidine recognized by a p-his binding domain? And what is the mechanistic basis of histidine phosphorylation? I've told you it can inhibit metal ion binding. That may be important, but we really don't know. And then in the last 5 minutes, let me talk about what we were really interested in studying with our antibodies, and that is cancer driven by aberrant histidine phosphorylation. So in liver cancer, we published a paper a few years ago now with Mike Hall's group, who developed a mouse model of hepatocellular carcinoma and found that NME1 and 2 proteins were highly upregulated and that the LHPP phosphatase was downregulated at the protein level. And so we collaborated with them to show that tumors from these mice compared to control liver pieces had elevated levels of 3 p-his proteins seen here, as well as higher levels of phospho-NME1 and 2 seen here. And they actually tested whether the loss of LHPP p-his phosphatase was important by using tail vein injection of an AAV vector to express LHPP in the liver of these mice, and showed this strongly suppressed the size and number of tumor nodules in these mice. They carried out p-his protein enrichment the same way we did and identified a number of potential proteins whose histidine phosphorylation increased, including ACLY, which I'll come back to in a minute. And then finally, they showed that in samples from human patients, hepatocellular carcinoma samples, there was also elevated histidine phosphorylation, as well as elevated NME1 and 2 phosphorylation. And as you can see, LHPP was strongly downregulated in these human samples. So this suggested maybe that LHPP is a tumor suppressor protein in liver cancer. So phosphohistidine ACLY could be important in disease. And potentially then one might be able to target histidine phosphorylation as a therapeutic approach, although I should say that NME1 and NME2 also have housekeeping functions, so that might be challenging. In pediatric neuroblastoma, we've worked with Pete Zage at UCSD and have published some of these data. So NME1 is and NME2 are on chromosome 17q, which is amplified in aggressive neuroblastoma, and patients with high levels of NME1 expression show poor prognosis. And Kevin Adam in my lab showed that cell lines, and this is not primary tumors now, human pediatric neuroblastoma cell lines have high levels of phosphohistidine, NME1 and 2 in this case, and total phosphoproteins in this case, phosphohistidine proteins. And these are the Boyle controls. And what Jacqueline Espérance in Pete Zage's lab showed was that if you knock down NME1 with shRNA, this accelerates the closure of a scratch wound, implicating NME1 in some properties of these cells. And Kevin Adam in my lab identified, and this was published, a number of proteins that can be pulled down with phosphohistidine antibodies and associate with NME1 by IPMS. So there are 30 proteins of this sort, and we are focusing on these 2 proteins, the prohibitins, which are known to have possible roles in invasion. These 2 proteins form ring-like structures with 16 to 20 subunits in the inner mitochondrial membrane, and it's known that PHB is important for OXPHOS. But there's also a cytoplasmic pool of PHBs that are associated with the plasma membrane, localized to invadosomes, which are ventral structures through which cells push through tissues. And prohibitin is a small protein, around 270 amino acids. It has only 3 histidines, so that's interesting. And here, Margot Sala in the lab showed that there is indeed co-localization of NME1 and PHB at membranes and in invadosomes. And she's also shown that one can co-precipitate PHB and NME1 from these neuroblastoma cells, and validated this by expressing the V5 tag form of PHB and shown that anti-V5 immunoprecipitation not only pulls down PHB, as you would expect, but also phosphohistidine containing NME1 and 2. So we have a hint that maybe PHB is actually a phosphohistidine binding domain that we're going to test now, which will be very interesting if it's true. In breast cancer, we've worked a little bit on LHPP, the phosphohistidine phosphatase, and this is the maybe my battery is running out here, so I may have to do this until the battery maybe recovers. What Jeffrey Riena and Kay Jung at UCSD have shown that the human breast cancer tumor samples have high levels of staining for LHPP. You can see a large increase here. You can also see a significant increase in these human tumors by immunoblotting. If we look at different breast cancer types, ER positive, ER, PR negative, HER2 positive, and triple negative breast cancer that lacks all 3 of these, it appears that it's the TNBC, triple negative breast cancers, that have the highest levels of LHPP. And in mouse models of TNBC and PY-MIL-T model, for instance, again, the tumor has high levels of LHPP staining that you can see here as well by immunoblotting. And in a very preliminary experiment using this MDA-MB231 cell triple negative breast cancer cell line that many people use, knockout of LHPP strongly reduces tumorigenicity in orthotopic mouse mammary xenografts in nude mice. We've repeated this once, so it looks like LHPP is pretty important for these tumors, and we've done some phosphohistidine protein analysis, but it's too preliminary really to show you. So this is ongoing work, maybe suggestive that in the breast cancer, LHPP is a tumor promoter rather than a tumor suppressor, which is often the way these things work. And then finally, and just a couple of slides on this and I'll stop, we've also used our expertise in pancreatic cancer, particularly in the mouse model, to study histidine phosphorylation. And I told you that Astar-CoA ATP citrate lyase might be important in liver cancer, but it's been shown to be important in pancreatic cancer by Katie Welland's group, who showed that if you conditionally knock out ACLY in the tumor cells in the KPC model, it strongly reduces tumor growth. And Astar-CoA synthase is a cytoplasmic enzyme that's critical for the generation of cytoplasmic Astar-CoA. And that Astar-CoA, as Katie Welland showed, is essential for histone acetylation. So that's her model. So Natalie Lutala in the lab used our SC44 3P his monoclonal antibody to blot KPC mouse PDA tumor tissue samples. And you can see very high levels of phospho-ACLY. Really not much change in phospho-succinyl-CoA synthase. So that was in keeping with Katie Welland's model, but she then developed a tyramide signal amplification method to stain tissue sections with immunohistochemistry. So she's done double staining with the 3-phosphorhistidine monoclonal and CK19, cytokeratin 19, as a tumor marker. And much to her surprise, most of the 3P His signal was not in the tumor cells, but rather in the stromal cells. So that's not what we would have predicted. And when she looked to see what stromal cells these were, it wasn't the cancer-associated fibroblasts. It turned out to be the CD45 positive cells, as you can see by this merge staining here. So she sorted the CD45 cells. So these are immune, I should have pointed out. CD45 is a marker of all immune cells, all immune lineage cells. And this signal appears to be largely due to macrophages and neutrophils, and it's mostly appears to be due to the phospho-ACLY. We don't have a sequence-specific phosphohistidine antibody. And so here is the blotting analysis of some of the sorted cells. And you can see there is phospho-ACLY. We're focusing on this band here, which is phospho-ACLY as confirmed by this dual color blot here. But what was most interesting was that if you look in immune cells, particularly in tumor-associated macrophages, there was this very strong phosphohistidine positive band not seen in tumor cells or in the fibroblasts. It is also positive for ACLY. This yellow band is both signals here. It's quantified over here. So it looks like there's a novel form of ACLY in macrophages. And that's something we're going to very actively pursue now to see what this might be doing in these macrophages in tumors. So stay tuned. So there's been huge success, as I told you, with developing tyrosine kinase inhibitors as a new class of cancer drug. And so what we're wondering is whether in cases where histidine phosphorylation is elevated in different types of tumor, whether we can find ways to target histidine phosphorylation, maybe hepatocellular carcinoma and neuroblastoma as a novel type of treatment. Now that's obviously a long way off, but it's an aspiration. So I'll just thank the people who did all the work. Yuxi and Ruijian Tian spearheaded the pancreatic cancer project, identifying LIF, with help from many collaborators at UCSD, UCLA, and the Salk Institute. My son Sean, together with Jennifer Cochran, did the LIF receptor trap, and we had funding from Stand Up to Cancer, which initiated the project at the Salk, the Lustgarten Foundation, and National Cancer Institute. And the work on histidine phosphorylation was driven by a fearless postdoc who took on this project without any evidence we'd ever be able to make these antibodies, but obviously was very successful, together with many people in my lab and UCSD and the tumor models with many different groups around the country and the world. And the mass spec was largely done, and structure work was largely done by two groups at the Scripps Institute in La Jolla, and I didn't talk about this work here. So thank you for your attention. I'm happy to answer any questions. By the way, in case you're wondering, this is the Salk Institute, and this is a piece of Chihuly artwork, glass artwork. This was on display during our 50th anniversary a few years ago. Yes. Thank you very much for that kind remark.
Okay, and my point is specifically regarding to the last part of the report. about this phosphorylation,
Yeah.
because as your group, several groups are working in that area and recently mentioning that this kind of modification is very important and critical in mammalian cells and in humans specifically. However, as you mentioned in a paper, the reported recently, this group mentioning that the proteins, the histidine phosphorylation is very complicated to determine for mass spectrometry. Practically, they made the experiment analyzing this kind of modification in human cells, and 90% of the histidine phosphorylation determined originally was not genuine from the point of view of this group. And this is not happening when they analyze or make analysis or studies with in prokaryotes, when it's clearly apparently that the phosphorylation in histidine remain in this kind of analysis using mass spectrometry. Do you think what happened specifically in this situation? Because this is important, because it established the controversy, and also groups is pointed that the phosphorylation in histidine is critical in bacteria, in prokaryotes, but maybe it's not so. It's not really. But in human is controversial, or no?
Right.
In your opinion, please.
I did say that it was controversial, and I asked you to read the paper from Simone Lemire's group in Nature Biotechnology to see if you agree with their criticism. We do have phosphohistidine peptides that do not contain serine or threonine, so we couldn't have misidentified it in those peptides because it was a phosphoserine. We have collaborated with Ron Moritz' group to really, and that's where this RMP M came out of. They reanalyzed our data and said they were convinced that we had identified a phosphohistidine site. So I think that the Lemire group, for one thing, they didn't find all the phosphohistidines they should have. There were several enzymes that we know they should have found that they didn't find. So clearly their method is not that good. I think, they found the most abundant phosphohistidine peptides, and they didn't see all of the more minor phosphohistidine. So they claimed that we got it wrong because our antibodies are not specific, and I think that's absolutely not right, because I've shown you the structures. We know they're specific. They do not bind any other phosphoamino acid that we've tested. And our blots suggest there is heat label phosphorylation of many proteins. They didn't really address any of that. But I agree, more needs to be done. So for any phosphohistidine site that has potentially been identified, one has to validate it by mutation to show the signal goes away, potentially make a phosphohistidine antibody, sequence-specific phosphohistidine antibody. So I think the jury's out. I mean, I'm convinced that it is important, but I agree that there are criticisms of what we did for the mass spec, not for anything else, but the mass spec analysis. So
thank you so much, and thanks again for your work.
Yeah, I agree. It's very important.
There are three questions in Zoom.
Yeah.
So two are from Dr. Esperanza Martinez, and the first one is LIF expressed in any normal tissue or in development?
So LIF is expressed in development. It is not an essential gene. So you can knock out LIF in mice and they are viable. The only essential function is in embryo implantation. So LIF negative, LIF null females are infertile. LIF null males are fertile. So it's not an essential gene, but it is, you can find it in normal. It is expressed, but it doesn't have an essential function. Now, on the other hand, the LIF receptor is essential. So LIF null mice die in embryogenesis. And that's probably because LIF receptor has another function. It's also a receptor for 4 other cytokines, but OSM and CTF1 and one other, so CNTF1 rather. So it's probably essential because it's working on other pathways.
And the second question is, will substrate availability such as serum circulating acetate or phosphate affect phosphorylation or acetylation?
Will the availability of substrates? Yes. Well, clearly it's known that if you deplete acetyl-CoA in cells, that histone acetylation is decreased, as Katie Well and others have shown. So acetyl-CoA can certainly be limiting. As far as I'm aware, there's no way you can limit ATP as a kinase substrate because cells try extremely hard to maintain ATP levels at a constant high level. So ATP concentration in cells, even when you stress them, is 3 millimolar. The Km for a protein kinase, including NME1, is usually around 10 to 20 micromolar. So there's always a huge excess of the ATP substrate for protein kinase. So I don't think it's going to be limiting for protein phosphorylation, but obviously in other cases, the methyl donors and things can be limiting for other post-translational modifications.
Otra pregunta aquí.
Thank you, Tony, for such a fantastic talk. I was wondering, in terms of the tumor suppressor function of LHPP in cancer, in liver and breast, is coming from the phosphohistidyl phosphatase activity, or is it possible that it has an additional role?
Right. So that's an excellent question, because the evidence that LHPP is really a protein phosphohistidine phosphatase is not that strong. There is a paper I pointed out in Molecular Psychiatry to you today where they claim LHPP dephosphorylates NME1 and NME2. We've not done a lot ourselves, but the structure of LHPP, the active site is capped by another domain. And it's been argued by Angelica Goka that that would restrict the size of the substrate. So we know it can dephosphorylate phosphohistidine. That's not in question. It's whether it can dephosphorylate a protein. And I think that's still a little bit up in the air. So one possibility is that the tumor suppressor function is not because it's acting on proteins, because it acts on some other substrate in the cell. But then you do have elevated phosphohistidine in proteins, but that may be independent of the LHPP loss. So I think it's still an open question as to what LHPP is actually doing and why it's different apparently in breast cancer and liver cancer.
And the second question, maybe I misunderstood, but did you enlist PP2A as a phosphohistidyl phosphatase?
Yeah, it's been reported. There are a couple of papers showing that PP2A can dephosphorylate phosphohistidine.
So how specific is the phosphatase activity?
Well, it's in, PP2A is very active. It was done in vitro. I don't know. I don't think the kinetics were very impressive for phosphohistidine dephosphorylation, but no. Phosphohistidine is primed to be dephosphorylated because the energy of the bond is so high that it's a good leaving group. So it's in mildly acid conditions, it just spontaneously dephosphorylates. So, you could argue that PP2A doesn't have to work very hard. So whether it's true in the cell, I don't know. But we listed it because that's been reported. So.
Thank you.
Sure.
So another question from Zoom is from Dr. Lopez Casillas at IFC UNAM in Mexico City. And he asks, why do plants need so many phosphoproteins?
Why do plants need so many phosphoproteins? Well, I think it's partly because they're stationary. So, this is the old argument, right? Plants can't move. So how do they avoid their predators, their pathogens? And one of the things they've done is to build this huge arsenal of receptor-like serine threonine kinases called RLK. So Arabidopsis has 1,000 genes that encode these. So these recognize PAMPs and pathogens and of all sorts. And they activate signaling pathways which then counteract the infection in various ways. So I think that's one simple answer, that they're using it as an immune defense system a lot. Of course, plants are just as complicated in terms of structure and cell types in many ways as mammals. And so they need a lot of kinases just to regulate cell function and lineage determination. And so but it's true, the phosphoproteome of plants is just as large as that of a mammalian cell.
Thanks. Well, I have a question about histidine phosphorylation too. And my question is, are there studies of this modification in archaea? Because in bacteria is well studied, and in mammals a little bit. I have this question.
Yeah, archaea obviously is a branch of life that is between bacteria and eukaryotes. I don't know of a study on the an experimental study on the phosphohistidine proteome that archaea have NME1 and NME2, because all branches of life need to rephosphorylate nucleoside diphosphate. So bacteria have an NME ortholog. It's called NPK, I think. But I don't know if anyone's actually looked at histidine phosphorylation in archaea. It will be very interesting, I agree, yeah.
Thanks.
Sure. I should say that bacteria use arginine, a subset of bacteria use arginine phosphorylation as a regulatory mechanism, and there's a arginine kinase and a phosphoarginine phosphatase. So whether that happens in eukaryotes in mammalian cells, I don't know. And phospholysine has been reported, but a kinase activity has been reported, but again, nothing is known.
¿Alguna otra pregunta? Si no es el caso, Tony, again, thank you very much.
Thank you very much for listening.